Unit 4 Chemistry, Chapter 16. So we looked at what the food molecules are. Now we're going to think about how we can break up those food molecules. So metabolism is when you have large organic molecules and you break them down into smaller subunits. Okay? It happens in your digestive system, and we need it to allow the nutrients to be absorbed, because essentially it's being taken in through very small holes in the villi, in the cells which surround your body. So big molecules like proteins or polysaccharides get broken down into their monomers. These reactions happen naturally, but so slowly. And because it happens so slowly, we need an enzyme to speed it up. And then once they get to the cells, those smaller molecules then get built up into larger, more complex ones, which is the other half of the metabolism process. Both parts are catalyzed by enzymes. They speed up reactions at the temperature that life exists at, so at your body temperature, for instance. And they can increase reaction rates hugely, by up to 10 to the power of 10 times. They're much more specific than chemical catalysts, so one enzyme will only catalyze one type of reaction. And they're so specific they even tell the difference between our optical isomers, so they wouldn't just do one or the other. There's very specifically to say just the cis or just the trans one. They have a spot on them called the active site, and the active site has got to be an exact fit for whatever the substrate is they're doing. And they are very heavily affected by temperature and pH. So whilst they can work within the conditions that life exists at, they also only work in those conditions. There are two different models of how they work. The first we'll look at, which you should already be familiar with, which is lock and key model of enzyme action. And I say you should be familiar with it because you should have studied this back in year 9 or 10, but you may well have forgotten it unless you do biology. So, the basic idea is that we have an active site which is the exact right shape for our substrate. So this is like the lock, and this is the key. It fits in to make the enzyme substrate complex. That then makes our product, and our products leaving your active site the same. Okay, so our groove is a concave area, and sometimes that's got a non-protein thing in it called a coenzyme, and we will come to look at that later. We hold the substrate in place by some chemical bonding, intermolecular bonding, so it's likely to be dispersion forces between nonpolar groups, ion dipole reactions, particularly if we've got a metal cofactor, because that's going to be in the form of a metal cation. We might have hydrogen bonds if we've got amine and carboxyl groups, for instance, or we might have ionic attractions. Okay? We've got to have the correct optical isomer, or these attractions don't occur, and therefore it doesn't fit. The other model is actually recognizing the fact that enzymes are a little bit squishy and they do move slightly. So what we're saying here is that it's not exactly an exact fit, but as it enters, the active site changes just a little bit. And that is actually important in the catalysis, because that can twist the substrate a little bit in order to weaken some bonds and get them able to break apart without having so much energy input. Okay? Same idea though, as soon as the products leave, we're then getting the active site back to its original shape. This one we'd call the induced fit model, because we induce it to fit a little bit. Now, at that active site, so this is our enzyme, our protein part, currently this one in particular is not active. We're going to add something to it which is non-protein. Might be a vitamin, might be a part of a metal cation, something like that. But we add it and it sits in this active site. That then makes our enzyme active, and it makes the cleft the correct shape, or perhaps with a bit of inducing, to our substrate. So one of the reasons why vitamins are so important in our diet is because quite often they are our coenzymes. So you may find, for instance, that the folate vitamin makes this coenzyme, tetrahydrofolate, and that's really important for transferring carbon groups. Others are important in redox reactions. Some move aldehyde groups around. Okay? There's a big wide range of what these vitamins actually do when they are acting as coenzymes.